So I'm going to be talking about Xenium. This is the particular data set is the 10x Xenium FFP lung cancer data set, and that's this one over here. You can find it on the 10x website under their freely accessible data sets. The actual full data set is over here. It's got like the output bundle, which is what I use to make a mini data set from. I'll show that in a second. And then if you this is just to show if you wanted to make to download the full data set yourself, you would need to get this particular file and also the OMI-TIFF and alignment file over here. But also just to flip over to the console for a quick second, something to do while I'm doing the introduction is probably just to download the mini data set. So I've made a mini data set that is available on Zenodo, and it is over here. It's the first chunk, I believe. Yep. So library, set the data paths, and then after that, the download file is here. So you would just run this entire chunk. I've already run it, and it's living currently on inside of my working directory. And it's just going to download this and then untar it. Anyways, back to the description of Xenium in this particular data set. So Xenium is 10x's in situ platform where you start with the slide very similar to kind of like Visium, except after you place the tissues, you first do a fix, and instead of also digesting that tissue somehow to get those probes to get the RNA to all fall out onto a capture lawn, with Xenium instead you have padlock probes that come out. So you have the RNAs that are living inside of your tissue. You have these padlock probes that are instead looking for your RNAs at the two ends over here. And then after it finds a particular match for the RNA it's looking for, it actually ligates across, and then it does rolling circle amplification. And that's how it forms like this tangle of DNA product that you now have inside of your tissue. So it's in situ rolling circle amplification. And now you have a very stable kind of signature for where this particular RNA originally was, this particular transcript. And you know which particular transcript it was because on the back of each of these probes, there is a barcode. And after having done this rolling circle amplification, you have a tangle that just has a bunch of these repeats of barcodes in it. And that means that if you then were to land some fluorescent probes on these, you would get a nice and bright signal, a very specific and very bright signal. So yeah, tissue placement, and then after that rolling circle amplification that goes on the machine, you do cyclical imaging, landing these probes onto that DNA nanoball you've generated, and then it does imaging in a cyclical process so that you get different signals for every single cycle. And then across different cycles, you get different flashes of light because of different fluorescent probes that are binding in sequence to your barcode. And then across different cycles, you get different flashes, and this is basically going to be decoded as individual like ons and offs that encode for a particular gene that you have in your design panel. So that's how this works. And also it works to give you a spatial resolution that's in the hundreds of nanometers. So it's very fine resolution, very highly resolved subcellular spatial data set. And this particular data set also was one of the first to showcase the recent multimodal segmentation outputs. There are 377 genes used in this panel, and the pipeline used is V2.0. It is now like 3.0 to account for the Xenium prime mainly, but most of the stuff should also be should also carry over, but I have to take a closer look at that first. I think I'll mention it later, but there is like one difference that I know of, but the convenience functions that we have also account for it. So the relevant items are, like I mentioned before, the Xenium output bundle and the supplemental H&E and the alignment file. So there's also some additional package requirements for running this particular tutorial. Main thing is that you will need Arrow, and it needs to be built with ZTSD support, which is the compression codec that 10x is using with these Parquet files. This is usually actually also built with whenever you use Arrow with Windows and Mac. It's just mainly if you're installing on Linux, I believe that you might have things that are optionally installed as far as compression codecs. So this is the expected directory output structure for a Xenium experiment. You have analysis, you have the auxiliary outputs, and the main things to focus on are the transcripts, the boundaries information, the matrix information, and also this is basically cell metadata with the cells.csv, .parquet, ZAR. And then we also will use the Xenium, the experiment.xenium file and the genepanel.json. So I'm going to go over what each of these, or like what we pull from each of these. But this is just to give an example of what we usually expect when we're running, or when we designed the convenience functions. This is the overall structure of the directory that we expect. And also it is important to note that they provide a lot of alternative formats. So that's very nice of them. And it's the same data that's contained within each of these, except I think for the boundaries information, in which case the boundaries for, if you're looking for the ZAR ones, have more information in them, but I'm not seeing this in this list for some reason. Maybe I forgot to add it. So the above directory is characteristic of Xenium v 2.0 pipelines. The only ones that might not be the same across outputs are the morphology focus directory. So that's this one, because you're usually going to get this one first, because this is the DAPI. This is basically an IF image for your DAPI. But these three over here are for individual other colors that you're going to use during the morphology staining process. So it's IF stains to tell you where your cell boundaries are. Yep, multimodal cell segmentation. And then as for the aligned images, so that's these two files over here. This is usually done after the Xenium experiment concludes, and it's added on using the Xenium Explorer software. The naming and location of these files are really entirely up to the end user, or the person who did the actual experiment and ran it. So it's probably not going to follow this naming scheme, or it might not even be in this particular directory. They might have to a separate way to hand it to you. So the mini Xenium dataset, which I mentioned earlier, is something I put together as a subset of the full object in order to speed up the steps here and make it hopefully locally runnable. This is a subset of the full dataset, which looks like this, by the way. And out of the entire area, which is roughly like 12,000 by, I don't know, 3,000 microns, we've shrunk that down to roughly 1,000 by, 1,000 by 1,500 or so. And this is the H&E aligned to the Xenium dataset with the micron scaling. So data preparation. Right now, we do require image conversion. So we actually have to first deal with the image formats before we touch the rest of the formats in the directory. Xenium generates OMI TIFF, which Giotto is currently not fully compatible with. So we have to convert them to normal TIFF images using OMI TIFF to TIFF. That's a utility function that we provide through Giotto class. It works through the Python TIFF file package. And the image files can then be loaded in downstream steps. So here's also some like additional code that's not needed for today because of the fact that I've already put together the mini dataset and done the conversion and also spatially aligned them. But the code that you would need to basically convert them from OMI TIFF into TIFF, this over here is just like listing the morphology focus images and also the H&E. What this is going to be a particular, this is going to be like a pattern search for only the image that we're using today. It might be named something else for you if it's a different like alignment image name. But it's looking for these image paths, and then after that, it's going to run OMI TIFF to TIFF across all of those, dump them into a single folder called TIFF exports by default. But I should also show that a little bit. So I have over here an example. Just want to put this up. This is the path to our directory containing the full data set. And I'm going to make also like a temporary directory just to store the images. So the image path, alignment path, and Xenium path. Um, as I mentioned earlier, OmiTIF, I'm just going to show the OmiTIF here for the post-Xenium alignment image. The OmiTIF here, the alignment CSV, which is basically like a matrix. I also show that later on, but so I don't have to show that later. I don't have to load it in with that read right now. And the experiment.xenium, which is basically like a JSON file that contains a lot of their Xenium platform settings. So once I have those file paths in, I'm going to run omiTIF to TIFF on this image path. The output directory is going to be to temp, and then I just have this here because I had something earlier. I don't necessarily need it now because it should be an empty directory. It should take just like a very short amount of time, maybe 30 seconds or so. Yeah, it's done. So the export image is now called the same thing, except without the omi.tiff. And just to show you what it looks like, I can load it in as a large image and plot that as. So it's pretty fast, even without the speed benefits that omi.tiff gives you, because it is doing like a sampling of the image to give you that speed. It pulls like regularly spaced pixels in order to represent the image. But yeah, this is what the alignment or the post-Xenium H&E looks like. You can notice that it's very much not aligned properly at the moment. So what we also have alongside that is a read 10x affine image. So this is not something you need with the normal convenience function. I'll talk through that later, but this is like if you wanted to just load in the image by itself and you didn't want to tell the rest of Giotto about where your file path is for the directory. It's for the output directory. For this function, you would need the file path to the image, the alignment path, which is the affine matrix path. Should just read that in. dt is equal to fread of align path. And that just looks like this. It's just an affine matrix. We'll also be explaining more about how affine matrices and affine transformations work on the third day. But yeah, here's just a preview of that. And then you'll also need the Xenium path, which is the experiment.xenium. That's basically a JSON-like file. So internally, we run like JSON-like read JSON of the Xenium path. And then it's all this information. The main thing that we actually want out of here, and actually I should include this as like the parameter name, is micron. That's the information we're trying to get out of here. So, well, I could just do this. This produces like a list-like structure, I believe. Oh, that's embarrassing. Okay. Pixel size, that's what it was. So this is the actual value we're trying to read in. If you don't know it, or if you didn't know it beforehand, you could have just like, if you did know it beforehand, you could just put in 0.2125 here instead. If you didn't want to actually look this up separately, you could just provide the path like what I had earlier. So that's this. Read it in, and it should now be actually aligned properly, like that. Another aspect to working with OmiTIFs that I just want to mention right now is that they also have a lot of OmiTIF metadata. It's an effort in the microscopy field to put together a very common set of put together a common set of universally recognized like metadata for how to read data, these images in, and how to like parse them. So it's a great effort to do it. We just need some catch up because of the way that we're working with images in Terra right now. But we're also working on approaches through TIFF file, like I mentioned earlier, to just be able to parse through the pyramids as well. Anyways, each of these OMI TIFFs come with a set of XML metadata. So this is just me reading in the image, one of the morphology focus images. This is the morphology image file path, morphology focus. And then after that, I am looking for the XML from it, and I ask for output as XML. So you can read through this using XML2 if you're familiar with that. We also provide other exports or other ways to parse through it. So if you have output as data frame, which is actually the default, it will provide you this. This is just me looking for channel information from the first items. From the first item in morphology focus, I'm looking for the first item in morphology focus, the metadata, and then I'm looking for the channel information that's attached. So that's what this is. And like I mentioned, the first one is going to be DAPI, and the other three are going to be segmentation aiding stains. So back to the mini data set and other things that don't require the full information to run. Again, I only did that on our cluster just because the files that I've included in the mini data set have already been processed to only TIFFs, and they have the XML information stripped away. So the files should have finished downloading now from the mini data set. I hope. Here is where I'm going to start talking about the convenience function itself. So it's actually seen some changes since the previous version of Giotto. We had a previous version of this function, but we've recently expanded it and are probably going to edit it slightly more in the near future to improve the flexibility. Right now, it's already it has most things except for like a parameter for instructions, which I did forget to add. But everything else should not be seeing any more changes. The main thing about it is that you can very simply, as long as you provide the path, directly generate a Xenium object, Xenium Giotto object. So again, this data path is pointing to the mini object I've made, which very closely reflects the actual both the file types and also the structure inside of this directory. And then I'm going to set the instructions for save directory and save plot. And then there's a lot of other parameters that were not used because it's just like a single line here. So I didn't use these parameters just now, but these can be very helpful if you have non-standard Xenium outputs for whatever reason. So right now, for example, expression path is just saying null, and so is like transcript path or bounds path. These have like very simplified or null inputs because if you have any alternative paths, you can directly set them here. And then it's going to find the correct look. It's going to find the information to read it directly instead of automatically checking for the expected directory structure. So the convenience function also auto detects the file paths based on the directory structure, and it looks for preferred file formats. So this convenience function will look for parquets for tabular information, and that's for like transcript data and the bounds path. So bounds is another word just for polygons in this context. The polygons and also the transcripts points information. Those are provided from 10x as parquets information. So we load parquets over instead of CSVs by default. It also loads the H5 for matrices instead of the .mtx or matrix market format. We also don't currently support ZAR, mainly just because there isn't great native R support yet for that. When you need a different file format or something, a different file format, like if you want us to use CSV instead, or something that's not in the expected output structure, you can supply a specific file path using one of these. So next, I'm just going to touch on the quality value. So this is something that Xenium provides as a measure of how confident it is about each of its calls for the transcript detections. So you can get a great idea of like how all this works also from the summary file, which I'm going to flip to for a sec. This is their summary file. It tells you like how many cells there are, the median transcripts. And then if you go to decoding, they have some information about their negative controls and also just how their quality values work. So you can see that lower quality values, there are some in this data set. They're mainly in areas where the tissue is not, and also in like certain tissue regions. But back to the script where I have like a condensed definition of a lot of those concepts. The quality value, the QV, is a thread-based, so 0 to 40 value that 10x provides for every detection in their transcripts output. A higher value equates to higher confidence in the decoded transcript's identity, and by default, 10x uses a cutoff of 20 for transcripts that they use downstream. If you ever set a value other than 20 for the convenience function through a QV threshold, then that means that the loaded data set will be different from what the 10x provided expression matrix and cell metadata is. Because they refer to values that pass the 20 cutoff as high, was it? High accuracy, high quality, yeah. As high quality transcript calls. And they basically use anything above 20 for the downstream stuff. So anything past 20 is the only items that ever go into their count matrices. So that's just one thing to keep in mind if you ever want to also load in the expression information on top of the subcellular points information, so that both those two pieces of information are compatible living inside the same Giotto object. And just some additional details about how the QV value is made is the raw Q score is based on how likely it is that an observed barcode that you saw previously with the different lights across multiple rounds of imaging, how likely that observed sequence is to be the code word, being the code word that actually maps to a particular gene, versus some other less likely code word. And then they further do an adjustment of that raw Q score that they've just found by binning the transcripts by Q value, and then adjusting the exact Q per bin based on the proportion of negative control code words that they detect within. And negative control code words are just one of the QC probes that they have in their panel. So they actually provide all of this different QC probe information inside the Parquet for transcript detections. And this is why we have this particular thing here for transcript type splitting. So yeah, feature type and also keyword as being the two parameters to take a look at for create Giotto Xenium object. You can see them right here. These are the defaults set for this particular pipeline version, so V2. V1 had a slight change, but the main thing is just that this allows you to split up your transcripts in a way that it's not going to interfere with your downstream analyses. You want all of your RNA or your gene expression probes to be separate from your QC probes so that they don't alter the way you do normalization and such. And this is just how you break up each of those code words or each of those different types of probes into different sections. I define feed type here as RNA, neg control probe, unassigned code word, and neg control code word. So those are the ones that 10x reports as different types of their QC probes. And then split keyword are just different patterns to match for inside of the naming of each of these types that you want to split for. So neg control probe maps to this one, unassigned code word maps to this one. It's as you would expect. And you can also have, if for whatever reason you have more than one item that you have, if there's ever more than one code word to match for, that maps to a single category. For example, if this is also blank, then you can add that as another item in here because this is a list. So V1 had a slightly different code word and also feed type that needs to be set that I'm going to detail more when I actually update the Giotto Suite website example. And also V3 has another included one, which is over here. So there's 4 types of transcript detections that 10x will report. Gene expression, like I mentioned, is the RNA gene detections. The negative control code word are code words that do not map to genes, but are in the code word itself. And this is just to show how specific their decoding algorithm is. So in all aspects, it's the same as any other barcode that might be used, except it doesn't actually point to a real gene. The negative control probe is for actual existing probes that do actual existing probes that are in the panel, but they target non-biologicals. So this is to determine how specific the assay itself, so how specific like the wet lab section of Xenium is. And then the unassigned code word are code words that should not be used in the current panel. They shouldn't actually be present in the panel. And then with V3 and their Xenium prime output specifically, there's also genomic control code words, and these are going to be probes that are for intergenic genomic DNA instead of actual transcripts. So the main thing to watch out for is that the other probes should be separated out from the gene expression or RNA feature type. And I basically described this already, where the split keywords are a list of character vectors that contain the grep terms to search for. And note that there's 4 feature types that I declare in the set of defaults. So 4 in here, but only 3 items in this list for split keyword. And that's because anything that is not matching to one of these is going to be dumped into the RNA section. All right, the first part of actual code to really run here, now that we've already made the Giotto object, is to calculate some spatial centroid locations. So I'm just going to run that. I'm going to calculate it for both the polygons for cells and also nucleus that we have. And then just to make a simple visualization to show that the data set actually works, we have this over here where I am now showing the cell polygons with the features. So just the first 6 features in the entire Giotto object. These are going to be like these particular ones over here. And you can see that they actually are mostly mapping to this area in here. And then something that we also provide as an alternative to the one-stop solution. The one-stop solution for importing data is the import Xenium. It's kind of like a lower level import utility that allows you to make independent sub-objects. If you're making create Giotto Xenium object, for using that, you're always going to get a Giotto object out of it, or either that or an error if something for whatever reason goes wrong. But with import Xenium, it actually allows you lower level access so that you can import only pieces or sub-objects from each of these data outputs that they have. So this is now a Giotto Xenium reader object. It's pointing to a particular directory. That's where I've saved the mini object. And then it shows you what the current QV cutoff is. It's set to 20. These are the file types that it's going to automate, or it's going to read by default, and then different functions that you can actually run in order to load individual pieces of data. So I can set a different QV. I would prefer to maybe like 10 if I wanted to have lower the threshold for calling high quality transcripts. And then if I wanted to load in the transcripts information, I could say tx is equal to x dollar load transcripts. And that's just like a single function call that should give me my transcripts information. And it says QV cutoff is now 10 for all of these. Also, something about the output here. The removed items here with 9,730 records or transcripts being removed out of the total, that's because we've set the QV to 10. If we set it even lower, that should be all of them that we load in. And you can see that this is now loaded in as a list of different Giotto points split by RNA, neg control probe, unassigned code word, neg control code word. So yeah, all of that's done by defaults, but these also have, as you would expect, additional parameters within if you, for some reason, needed to change them. And you can also set a specific path instead of the default one that's baked in to this function by now. So you can plot this. Oh, I have the actual function here, or the actual call here. You can plot this as density to get an idea of that. And then, yeah, this is just to show the contents of the Giotto points object. I can also load in the polygons or the cell by default, and that's what you get. So I am It's optional, but you can also remove transcripts at this point in order to save space. I'm just going to keep it for now, though. Next is showing how to actually edit the file type that you load. So there's three slots here that you can edit directly. So you can change which Xenium directory you're pointing towards. You can change the QV threshold. You can also change the file type that you're looking for. So you can further index into here and find that there's entries for cell metadata, expression information, boundaries, and transcripts. So here you can change which particular file type is going to read by default. So here I want to change the expression information to read MTX file format instead. And you can see that that gets reflected inside the show function. It's now MTX where it started as H5. And I'm doing that because H5 is not in the data set, not in the mini data set. The one I exported was in MTX format instead. So I'm just going to load in the expression information. And that looks like this. It's a Giotto expression object that is also split across the different feature types. That 10x reports. So this one's for RNA. This one is for negative control probe. And control code word. And unassigned code word. You'll notice that also that these names are a little bit different than the naming I set earlier. You can access that by looking at feed type EX, feed type of the expression information, which works across lists. So these are the feature types of each of those expression objects. They have spaces in them, which is not ideal, and it's a little bit hard to work with them. So we are going to just change them into the same types of feature types that we mentioned earlier for the transcripts. So I'm going to set them like so. And now they actually have the correct feature types. And because these are already like Giotto sub-objects, you can append them directly into the Giotto object in a single step. I'm going to make G2 first, though, just because I don't want to add the expression information into the main one that we're going to be continuing with for downstream purposes. We're going to be using Giotto's own aggregation method with the overlaps to make the expression information instead of using 10x's. Set that, and you see a descriptive message for it. And I should also load in the cell metadata. So I'm loading that in the CX, which looks like this. You have the cell IDs, the transcript counts, control probe counts. So all the QC information, total counts, cell area, nucleus area. And then you can set that into the Giotto object or G2 as well. And now you have that information available to you as aggregate data. And just to show that we can actually plot from these 10x provided metadata information. I'm going to plot the cell area. So this is a plot of cell area for each of these polygons. And then this is going to be by transcript counts instead. Very similar as you would, expect. And then I'm just going to remove G2 just to save on space. Where am I? Okay. So back to the images. We've created the Giotto object itself already, which is in G, that has our subcellular information and a set of spatial centroids that we've calculated. But we haven't added the images yet. So just to go over the different types of images you'll likely see, the morphology omiTIFF is something that's at the root or like the root level of the directory outputs. This is a Z stacked image of the DAPI staining with Z levels separated as pages inside of the omiTIFF. So it's like you have individual images that you can load in each for different Z levels of a confocal imaging stack. I believe there's like 11 pages or 11 images inside of there, but most of them are blurry actually. So it's only really pages 6 and 7 that are very in focus. And this is only with DAPI staining. So I'm not going to load this in. We also don't really have a good way of dealing with 3D images yet at the moment. But I am going to load in instead the morphology focus images, which is a folder that contains single channel images, but with the original Z information collapsed into a single in focus layer. So for all datasets, image 0 will be DAPI staining, but if you have additional stains, which we do here because of the multimodal segmentation, they'll also be here. And these are the recommended IF images to import. Xenium V1 human lung cancer, the OMI-TIFF image, which is the add-on. This is the H&E that we're going to add on with the affine that I showed earlier. So there's also the image metadata. And I touched on all of this previously. So that's the function I ran to get the metadata from one of the IF images. I did actually also add in the OMI XML as part of the mini dataset export. You can load that as this file path. And this is just to show you the internal XML2 code that would make it possible for you to parse through that XML information to get the exact same output that we saw earlier. The only reason why it's done this way instead of with OMI-TIFF metadata, as shown earlier, is because that function actually does require an OMI-TIFF to work on, whereas this is just directly working with the XML file itself. Okay, so the morphology focus images will need to be scaled by the micron scaling factor. The aligned images need to be first affine transformed, then scaled. The pixel size is 0.2125. And then I think I have this on the actual, this is just to give an example, understanding of what the images look like versus the vector data. So the vector data itself, which is already scaled to 1 micron. So this is, I think, actually the spatial size of all of the transcripts information. And then you have the IF images, which are in blue over here. And then in gold over here, you have the starting orientation for the H&E image. So all of these need to be mapped back down to the red portion over here. And these are also the convenience function parameters that you would use for loading in images and the importer parameters that you would use. It's the parameters called like load images, and you would provide a list of different names for the images you want to load and also the file paths. And then that automatically does the micron scaling for you. And for the aligned images, you provide a list, a named list. So the name will be applied to your image. And then after that, you also supply a first up path to the image and then a path to the alignment CSV. And the importer params are shown here. So this is the import Xenium function instead of the create Giotto Xenium object, where you can say X load image. Well, I can't do it here because I don't have OMI-TIFFs, or because I don't have the original raw locations of these images. You would apply first the path and the name. And then same with the aligned image itself, but you'd also supply the alignment path. And I mentioned read 10x affine image earlier with the demo. So I'm just going to load in the actual mini object images now. So these are the actual image paths. The image list I'm going to create using those paths is this, where I've also named it based on the channel names that I got out of the XML. And I'm going to say use rast extent true just because that's, if you ever have images with tagged spatial information, you would say true for that. And then that puts it in the correct place spatially. So I've loaded in these 5 images, the DAPI, 18S, etc., also the aligned image. And then just to make some of these images brighter, just because they're currently a little bit dark, as you can see with image list, the first item, it's completely dark just because it's scaled to a certain like bit depth. However, the actual values encoded are kind of low. So to get around that, we can set a max window. of 5,000 for each of these, and that basically sets the image scaling for them. And I'm going to set them in. And I can also show that now that I've set the window, it should be a lot brighter. So this is the DAPI image. You can also check on this kind of stuff by going to dist image, I believe. Dist Giotto image, yeah. This gives you like a distribution of the values of inside of your image. Giotto large image is equal to your image list. And then this is just the distribution of the values that you have in your image, with the red line being where you've just set the max window. So this is, we're going to ignore any values past here. And when we're showing this in a plot, it's only going to scale all of your RGB values across this range. So that's why it appears brighter now. So I've just added those images to the Giotto object, and now I can do a spat in situ plot to just basically show a couple of these images along with some polygon combinations. So this is showing the H&E image mapped to the rest of your data. So this is the polygon cells in cyan. And this is the DAPI with nuclear annotations to show that they actually map quite well. 18S staining with the cell annotations, and then ATP, CD4, and cadherin with the nuclear annotations. Okay, so moving on, spatial aggregation. This is where we calculate the actual transcripts that are covered by each of these polygons. We're using cell here to overlap the RNA transcript, the gene expression transcripts. And that's going to turn into, in our next step with overlap to matrix, it's going to turn into the cell by feature count matrices. I'm going to run that. And then we can just basically do the aggregate analyses workflow that you've already seen a couple times now from previous sessions. First, though, I'm going to show maybe a little bit of a gotcha with Giotto. It's that if you want to add statistics to get an idea of the raw expression information, right now it's still going to say you haven't done the normalized expression first, but you don't actually have to do that. You can actually tell it specifically which expression you want to use first. You don't have to use only that one. And then I can pull, using pdata.dt, the cell statistics, and that is now stats of your raw information. So cell ID, number of different features discovered inside of this cell, percentage features, and then the total expression counts that you found in here. And now that I have that, I can actually do a quick ggplot histogram to show you the number of counts detected, or sorry, total expression counts per cell, with these being like the counts of cells with this particular amount of total expression. And these are with bin width of 5 right now. So every single one of these columns means like an increase of 5 in terms of total expression. And so I'm going to start with some filtering on this Giotto object. I'm going to set very permissive filtering, mainly just for removing any 0 values from the matrix. The reason why I do this is just because differently from single cell, and also situations in which you just have a large amount of genes and calling that may or may not be accurate. This particular assay with Xenium, you have pretty accurate cell segmentation. So if you get a particular cell, you're pretty sure that it is actually a cell. And also with the transcript detection, transcript calls, they're usually pretty highly accurate. So I'm going to set some very permissive thresholds of just like 1. You only need like 1 feature detected per cell for it to be, or sorry, you only need a feature to be called once in order for it to be kept. And then you need like 5 features per cell. So anything in this first bar is going to be cut out of the data set. And that removes 143 out of 7,655. and then normalize that and add statistics, which overwrites the previous raw statistics with normalized statistics. And I'm just going to plot number of features detected per number of unique feature species detected per cell and total expression. And then moving on to dimension reduction. So basic aggregate workflow. I'm going to run the PCA. I'm going to set features to null. And I'm setting features to null here just because there's no HVFs that I'm actually using. Normally, it's a very good idea to use HVFs so that we can decrease the time that run PCA takes, and also so that we can only focus on the actually highly variable features, like Jocelyn mentioned, that greatly drive how different cell populations are different from each other. But here, since we are only starting with like 100 or 377 different genes, that we really need all that we have. So this is the scree plot. We calculated 100 different PCs by default, but we're showing up to 30th. And then I'm going to run a UMAP on this, which is going to take a second. Going to use all up to the 15th because that looks like a good cutoff for the amount of variance that is explaining. And then I'm only using like the number of neighbors, or I'm just showing here that the number of neighbors I'm using here is 40 because that's the default value. Plot PCA. That's how our PCA looks. The UMAP is actually already quite well structured as well, and that's possibly related to how good the segmentation is for this data set. And I'm just also going to make a nearest neighbor and do nearest neighbor network and then create lighting clustering with the nearest network covering all 15 dimensions that we calculated for the PCA, the UMAP earlier. Make that similar. And plot 3D of the UMAP, or sorry, of the PCA. So previously the PCA was, I'm just going to flip back to it for a sec. The PCA looked a little bit hard to figure out because it's like 3 random pieces. If you were to look at this with the same 2D projection, it's going to be a lot more confusing looking, but you can actually see that it's like, it's got some structure to it in 3D as well. And UMAP plot is also well clustered, so it maps quite well. And that spat plot that, or spat in situ plot this with the line clustering annotations. You can see that it's quite well spatially patterned. So from here, we can also do some niche clustering. So we can build on top of the line annotations and define spatial niche structures for the niches, meaning like situations in which Leiden types are often found in similar concentrations or amounts. So in order to do that, we first have to set up a spatial network so that we can figure out the spatial relationships between the cells. I'm going to set one up with Delaunay and just plot it. That's what that looks like. And then I'm going to run the niche calculation, which I think Ruben already showed earlier. We basically calculate based on the percentages of line cluster annotations, both for the cell itself and also its surrounding neighbors. And then we're going to add that as a new column inside the metadata as light and niche. And then I'm going to do retrieve the niche information. Oh, sorry, it's not in metadata, it was in the spatial enrichments. So the prop table itself looks like this. So by cell IDs, and then each of these are the different niches. Or sorry, these are different line clusters. And then each of these values are basically like how much signature that particular cell and its surroundings has for this particular lighting cluster annotation. So next, I'm going to take that property table. I'm going to convert it into a matrix. And then I'm going to set a seed so that it's not randomized. And I'm going to run k-means in order to decide, or I already did all this, in order to decide a single cluster for these different niches to mainly be categorized as. So that's what this is over here. And then we can also visualize the niche makeup. So that's basically this set of annotations, except also plotted with the coloring scheme used for the line and annotations. This shows you the different niches and then also which particular line and annotations make up each of the niches. And next is cell proximity enrichment. So this may take up a lot of memory, but we're using the spatial network again. Just going to start running it. It determines if there's an enrichment or depletion between annotation types by calculating the observed over the expected frequency of interactions. Okay, there it goes. So what this provides is a set of interactions where these numbers are basically your lighting, and these are your lighting annotations. And if it connects to something else, that's another cell. Or this is how commonly you see cell interactions, cells being next to other cells with the same annotation for 1010. Same for 6,6. So these are basically like between similar types. You would expect that these are usually pretty high because your clusters are spatially organized. So over here is just enrichment of these interactions versus depletion. And then also over here, you have the number of interactions that are actually existing for each of these situations. So rows where you have a lot of interactions are more likely meaningful, and also where you have a large amount of enrichment or depletion. Particularly interesting probably are like 6 to 8 and 2 to 9, just because these are non-homo interactions between basically like non-self-to-self interactions. It's like self to other cluster annotations that are pretty common actually. So I can just directly, with that understanding, set up a couple of color bars or color scales and plot this in situ to show those two particular lighting clusters and see that they are indeed commonly found next to each other. So that was first 6 to 8, and then this is now 2 to 9. Another thing that we can do with this data set is generate pseudo-Visium data sets by tessellating across it with the same layout and resolution as a Visium capture array. So we have a function actually for this that's called make pseudo-Visium. And that will generate a set of circular polygons across the spatial extent that you provided. And here we're also using extent right here with the prefer argument pointing to polygons and points. And all data is true. So this is basically looking at the Giotto object itself and seeing what spatial region it occupies. And this is looking at both the polygons and also the points information in the object. And because it's saying all data true, that means that it's not just the extent of the polygons and the points, which may be slightly different from each other, it's the combined extent. So anything that pushes on the bounds of that spatial envelope basically is calculated into here. And micron size is 1 because that's actually the scale that Xenium data is scaled to by default. So I'm just going to make pviz, and that generates 76 polygons. I'm going to add this to the Giotto object and calculate the centroids again. And just to plot it, these are the polygons that we just added to the object. And then we've just added pseudo-Visium as the polygons. So we want to be doing our next steps with it. And we're going to set our active spatial unit to that. We can calculate overlaps, find the matrix, or calculate the matrix from it, and then filter it quickly. So this time we're using less lenient filtering. And then we're going to normalize, add statistics, and plot it. So I just breezed through most of that because it's stuff we've been through already. And then this is just the total expression aggregate under each of these pseudo-Visium spots. So then I'm going to also run PCA, UMAP, and here's network and clustering, this time with a resolution of 1.5. And just to show you the output of that, this is the PCA of the pseudo-Visium, the UMAP of it, and then the spatial plot. And that's what I have.